Hello students, welcome to Molecular Biology part 15. The concept is translation. Protein synthesis is directed by DNA, which is confined to the chromosomes in the nucleus. The synthesis of proteins occurs on the ribosomes in the cytoplasm. So already we discussed during transcription in prokaryotes as well as in eukaryotes. So DNA having the information about the sequences of amino acids in a polypeptide chain which is present in protein. So that information brought by mRNA to the ribosomes, and in ribosomes the protein synthesis will takes place. So this concept at a transcription we are discussed. So now the protein synthesis is the action of a gene, or the ultimate expression or action of a gene is a protein synthesis. So that gene directed to synthesize these proteins, and they are located in chromosomes in the nucleus. From there they are directed to synthesize proteins in ribosomes through various RNAs. Now, various steps involved in protein synthesis are: 1. Attachment of mRNA with ribosomes and the formation of initiation complex. So, the first step in protein synthesis is the formation of initiation complex. How it forms? It forms by attachment of mRNA with ribosome, forms that initiation complex. And a second step is activation of amino acids and their attachment to the tRNA, transverse RNA. Third one, elongation of polypeptide chain. And fourth, termination and release of polypeptide chain. These are the four steps, and these four steps we are going to discuss in this video. Now the first step in the translation: attachment of mRNA with ribosomes and the formation of initiation complex. Before protein synthesis, the ribosomes occur in the dissociated and inactivate state. So already we discussed in previous video two concepts. One is TNA, tRNA, a adapter molecule, adapter molecule. Along with that, another one, structure of ribosome. Structure of ribosomes are also we discussed in the previous video. In the structure of ribosomes, we discussed that the ribosomes are made up of two subunits, small and large subunits. Small and large subunits, and these subunits are dissociated and in inactive state. That means the subunits are present separately. They are not united before protein synthesis. So before protein synthesis, ribosomes, the subunits of ribosomes occur in inactive state. That means in dissociate form. They are not associated together. For the formation of initiation complex, three factors need to be present. Let's see what are the three factors need to be present. These are the three factors are one, mRNA with the AUG codon as the starting or the initiation codon. That is a first factor. And second factor, first tRNA, transverse RNA, that is methionine charged tRNA. The first tRNA is methionine charged tRNA carrying anticodon, that is UAC, to match with codon AUG. So the first tRNA having anticodon, already we studied that adapter molecule, one end, third end, it consists a site for amino acid attachment, and another end it has the anticodon loop or anticodon region. That anticodon is opposite to the or complementary to the mRNA. So now in first factor mRNA with AUG, then automatically the complementary codon present on anticodon is UAC to match with that codon present on mRNA AUG. Third one, both the small and large subunits of ribosomes need to present. So all these three factors are essential for the initiation of this protein synthesis. As UAC anticodon of tRNA matches with the first triple code or triplet AUG of mRNA, the smaller subunit of ribosomes joins with the complex in the presence of magnesium ion. Immediately later, larger subunit joins in the complex. So once again I am repeating, as UAC anticodon of tRNA matches with the first triplet code or triplet code of mRNA, that is AUG. When it matches, anticodon is matches with a codon of mRNA. When it matches, because presence of AUG is the initiation codon. When it present the initiation codon and each matches anticodon on tRNA, then the smaller subunit of ribosome joins the complex in the presence of magnesium ion. Immediately later, a large subunit joins in. Now, the intact ribosome is formed and is known as initiation factor. So here now intact, that means associated ribosome is formed intact without damage. That means unseparated, non-separated ribosome subunits are formed is known as initiation factor. The methionine tRNA in bacteria, the initial methionine carries a formyl group, hence it is known as N-formyl methionine tRNA in prokaryotes. But in eukaryotes, it is called as methionine tRNA. But in bacteria, the initial methionine that we will consider as initial tRNA is N-formyl methionine tRNA that binds at the initiation site is different from the tRNA. The tRNA, the methionine tRNA that binds at the initiation site is different from the tRNA. So this is the representation what we discussed in previous slides. So now the first mRNA. Now when the mRNA having a triplet codon, that is AUG, when it matches with that tRNA, it is a tRNA molecule. tRNA matches a anticodon, that is UAC, then it initiates the process. When it matches the 40S, that means in eukaryotes 60S is the largest subunit and 40S is the smaller subunit. The smaller subunit is joins in the complex, and this we will consider as the initiation codon. Now the tRNA with this methionine, then it is called as the methionine tRNA. So the short form of this is tRNA met. So we will write like this also. This indicates that methionine methionine tRNA. And now later it joins a large subunit to this complex, then it forms as a intact ribosome. That means undamaged. So before we are given point that before protein synthesis, ribosome is dissociated and inactive state. Dissociates into two subunits, larger and smaller subunits. And during this translation process, then it joins to form intact ribosome to translate and to synthesize the protein. There are two tRNA binding sites on each ribosome. A, A or amino acyl site. It binds the incoming amino acyl tRNA, the tRNA carrying the next amino acid to be added to the growing polypeptide chain. Okay. So now first site on this tRNA having two sites. In that first one is amino acyl site. So now this amino acyl site binds the incoming amino acid, next amino acid. That means we said that polypeptide chain formed with many amino acids. So amino acid 1 binds with amino acid 2, binds with amino acid 3, like that. This is like n number of amino acids are present. So now in this chain, next amino acid to be added to the polynucleotide chain to grow. So now how it added? By the presence of this site, that is amino acid tRNA site, the incoming amino acid carrying the next amino acid to be added. So the amino acid tRNA brings the next amino acid to add this polypeptide chain to grow its length or maybe size of that polypeptide chain. And second one is polypeptidal site. So this polypeptidal site binds the tRNA to which the growing polypeptide is attached. So now let's see the diagrammatic representation. So starting codon AUG and the tRNA with this UAC, that is it is tRNA or initiated tRNA. It is matches with the codon of mRNA. So this is mRNA strand having initiation codon or start codon, and the tRNA matches to that and it is initiated. Soon after matching of this tRNA with this mRNA codon, then subunit, smaller subunit is joins. This green color one is smaller subunit. And see here the initiation mentioned with F, formyl methionine. That means this representation belongs to prokaryote because prokaryotes are having formyl methionine tRNA. So it is formyl methionine tRNA. It is occurs in prokaryotes. So here the smaller subunit is 30S. It is a 30S subunit is joins in the initiation complex. And then soon after, immediately later, the larger subunit, that is 50S subunit, is joins to this complex and it form as a initiation complex that we discussed the first. So this is overall representation of that initiation, formation of initiation complex. So overall, so the first one, the initiator. Here we are mentioned with only methionine. That means this diagrammatic representation, it is eukaryotes. Eukaryotes translation. If F is there, formyl is present, then it is. It is a methionine tRNA and it is matches with the start codon, and a smaller ribosomal subunit is also joins to this codon. Now the initiator codon, when it is joins with that methionine is formed, and later immediately larger subunit. So here the smaller subunit is 40S because of eukaryotes, and after joining this smaller subunit to the complex, then immediately larger subunit, that is 60S, is also joins to form initiation complex. So that means it become intact ribosome. So ribosome with larger and smaller subunits. Activation of amino acids and attachment to tRNA. So how the amino acid is activated. So each of the 20 amino acids occur in the cytoplasm in the inactive state. 20 amino acids occur in inactive state. The specific activating enzyme known as amino acyl tRNA synthetase enzyme, amino acyl tRNA synthetase enzyme, and ATP molecule react with the amino acid, result in an amino acyl enzyme complex. So the first step is amino acid reacts with or activating by an enzyme. So E for in this equation, E is the enzyme, that is amino acyl tRNA synthetase enzyme along with ATP energy molecule in presence of magnesium ion to form amino acyl enzyme complex. When it utilizes ATP and it forms pyrophosphates, two phosphates are released. So enzyme complex AMP, that is adenine monophosphate. So adenine triphosphate become in the complex monophosphate. Two phosphates are released out. So we need to remember amino acid is activated by an enzyme amino acyl tRNA synthetase and ATP molecule. When it is utilizes, the amino acid form as a amino acid along with this enzyme form as a complex, that is amino acyl enzyme complex. What is present in this amino acyl enzyme complex? Amino acid and enzyme together formed this complex by using ATP. The amino acid thus formed is said to be activated. So that is amino acyl enzyme complex is said to be activated. The amino acid, activated amino acid, remains with the specific tRNA molecule and one of the loops of tRNA recognizes this tRNA synthetase enzyme. We are given T loop recognize the tRNA synthetase, that means amino acyl tRNA synthetase enzyme is recognized because amino acid is active by reacting with that enzyme. The enzyme till attaches to that activated amino acid. So enzyme with active amino acid. So the T loop of adapter molecule, tRNA molecule, identifies the active amino acid by that amino acyl tRNA synthetase enzyme. Now, the amino acid is transferred to the amino acid attachment site of tRNA and enzyme is said to be free. Amino acid attachment site is third end. Third end is the amino acid attachment site and enzyme become free. The resultant compound is amino acid tRNA complex. So now amino acyl enzyme complex, due to presence of this enzyme complex, due to presence of this enzyme complex, presence of this enzyme complex, the one of the loop of tRNA identify the active amino acid. One amino acid is active, one amino acid is reactive with an enzyme, amino acyl tRNA synthetase enzyme. So when it reacts with that enzyme, the enzyme and active amino acid together present. So when the enzyme is detected, then the active amino acid is attached to the attachment site of tRNA, and amino acyl tRNA synthetase enzyme is separated, or it become free. Again, this is repeated. Again, this amino acyl tRNA synthetase is reactive with another inactive amino acid and make it as active. Okay. So whatever given in this part, that is given in the equation form. So amino acyl enzyme complex is a react with the tRNA, attached with tRNA. So tRNA takes this amino acid, then it releases that E, here E for enzyme, it releases the amino acyl tRNA synthetase enzyme. So E for enzyme. Enzyme is released, and this enzyme again reacts with another inactive amino acid, and this become AMP, adenosine monophosphate. So it reacts with a phosphate molecule, and it forms first ADP, and then it is also reacts with another phosphate to become ATP for further usage. Third step in translation, that is elongation of polypeptide chain. In both prokaryotes and eukaryotes, the process of elongation is basically the same, and the elongation factors are required at various steps in the process. Elongation factors are required at various steps in the process. The addition of each amino acid to the growing polypeptide occurs in three steps, which are followed in a cyclic fashion during elongation process. Step 1. Binding of an amino acyl tRNA to the A site of ribosome. A site of ribosome. What is that A site? A site is the codon, triplet codon on mRNA, and the matching to this anticodon. That region we will call as the A site. Okay. So matching one, and we will call as A site. So binding of amino acyl tRNA to the A site of the ribosome. The tRNA binds to the mRNA by forming a complementary base pair with the tRNA anticodon. So we know that. So these codons are called as this codon. You may see that uh, these codons are called as A triple AAA. It is called as anticodon because the codon present on tRNA is called as anticodons. This is the anticodon. Anticodon is present on tRNA. So it is a tRNA molecule having anticodon, and this strand is mRNA, and the code present on this region are called as the triplet codon. So this code based on the DNA. These codes are copied from the DNA. So now based on that, the tRNA binds to the mRNA by forming a complementary base pair, C triple U. The complementary base pair of this anticodon is AAA. is the complementary. And similarly, next UCG, AGC, it is anticodon. UCG is a triplet codon on mRNA. Pair to this with the tRNA anticodon. So based on this, here it is triple A. Then the amino acid. That means here the loops are identified. That enzyme activated amino acid with enzyme. That amino acid is attached to the third end. So based on this anticodon, and here the tRNA, that means the loop, T loop or T arm of this tRNA identify the active amino acid and it is attached to this end. And when both are joined together, it form here see yellow shade is given here. The bond formed between two amino acids. This is peptide bond formation. Then after formation of that, this one moves out to that we will call as the exit. So that is P site for peptidal site. That is that we are mentioned it two main sites. Amino acyl site, that is amino acid adding site, and peptidal site. So P, it is a P and amino acyl, that means the two sites the bond is formed. Then from this P it is moves to the existing, that is the E site. So a ribosomes having actually having three, that is amino acyl site and peptidal site as well as existing site. So the tRNA will comes out and again it brings one more anticodon. That means tRNA comes with a matching or complementary codon and it collects the amino acid and it forms the bond. Step 2. Transfer of the growing polypeptide chain from P site, peptidal site, to the A site, that is amino acyl site, by the formation of new peptide bond. The elongating chain from tRNA in the peptidal site is uncoupled and covalently joins the chain to the tRNA in the A site, amino acyl site. This reaction is catalyzed by the enzymes called as polypeptidal transferase enzyme, which is present in the 23S RNA of 50S subunit. Formation of peptide bond between the amino group, that is NH2, between amino group of amino acyl tRNA in the A site, amine group in the A site, and carboxyl terminus, carboxyl, that is COOH. This is a carboxyl terminal, carboxyl group of the growing polypeptide chain attaches to the tRNA in the phosphate site. The peptide bond formation requires ATP, GTP, that is guanosine triphosphate. Hydrolysis, that means by using of this GTP, the peptide bond formation occurs between the two amino acids. Let's see the diagrammatic representation. So now this is a P site and A site present in the initiation complex, the complex of this. So now A site, it is based on the mRNA, tRNA molecule having anticodon to complementary to the codon of mRNA. Based on this codon, here the tRNA having a loop, D loop as well as T loop. The T loop identify the enzyme amino acid tRNA synthetase enzyme which is attaches to the active amino acid. So based on that, it brings the amino acid. So this circle is amino acid. Okay. So now when it brings another amino acid, now this tRNA shifted to this P site and A site is become vacant. Then again tRNA brings another amino acid. So here and it these two amino acids one and these two it is shifted to P site no and again new N for new amino acid bring by tRNA onto the A site. So bringing this it is polymerized, that means the bond formed between this amino acid. So this amino acid, this side end, left side end having N terminus, that is amine group terminus, where in A site, amine group, and in P site right hand terminus, right hand side of amino acid molecule consists carboxyl group is present on the right side. So the peptide bond formed between these two groups and it is characterized by the peptidal transferase enzyme. Okay. So that peptidal transferase enzyme is present in the 23S ribosome unit, RNA 23S rRNA of 50S subunit. That unit consists this enzyme. Okay, the bond is formed. Then what happens? It need to extend from this P site after forming this. Then this amino acid will comes out. That means extend, and this is sent out. That is called as the E site. Through E site, it existed from this P site. From the P site, the tRNA, first tRNA is existed. Then the second A site, already second is when it is existed, this mRNA or this amino acid pull to this P site. Then A site become vacant. So I think you are following. So when when the tRNA after formation of bond, the first one, this is a first and this is a second, a new one. This bond formed between first amino acid and second amino acid. Then after that, the first tRNA molecule or first amino acid which is brought by the tRNA that is existed from that P site and moves to this A site region. It comes. Then when it is existed, then it pulls this A site. That means the mRNA present on this A site is pulls onto the P site and A become vacant. A become vacant. Then the third amino acid will joins on that A again. The same process, complementary to the complementary to this triplet codon mRNA. Based on that, again one more tRNA will comes and joins on the A site, and again bond formed between third amino acid and second amino acid. This is 1, 2, and this 3. So like that it continues. Once the bond is forms, then again the P site, from this P site tRNA existed. Then from that, here the third tRNA, it will move on to the P site. Then it become vacant. Then fourth amino acid will comes on that. Same repeated. Like that several amino acid till format till appearance of stop codon, it will continue. the extending or growing of polypeptide chain will continue. See in this representation again. See, it is named as NH2 amine group, and it is E site and P site is mentioning. Then this is the A site. It is vacant. When first two are bound together, then it will pull onto the E site, and second one comes onto the P site, and then the A site become vacant. So the next charged tRNA binds to the A site. So it is having TRP, tryptophan. So here it is phenylalanine. P site having amino acid phenylalanine. Now that A site, see in the second diagram, that is A site, it is the third tRNA, it is comes with the tryptophan amino acid. So this tryptophan bonds with the phenylalanine. So this side end having amine group, and here this A side, this amino acid having this region having amine group here, and phenylalanine carboxyl group joined together to form peptide bond. So then what happens after that? It will comes existed. Then it will comes to the, it is existed now. See in this diagram, AAG, that is P site. What is represent? Phenylalanine. It comes into the existed. That means here it is amino acid is not there. Over. It is entered into the polypeptide chain. That means the P site is comes on to the existed. And this tryptophan amino acid molecule is comes on to the P site. So it is E and it is P. Again, next codon CAG. This codon is become site A. Based on this, tRNA again brings another amino acid, that is GUC. So GUC codon comes onto the A site, and again that GUC represent amino acid that bonds with this tryptophan. So whenever the peptide bond is formed, then this tRNA molecule, it attaches to amino, it becomes the attached amino acid is joins with the polypeptide chain. So then it become without amino acid. That's why it is amino acid free tRNA. Then it will comes out from that site, P site, through existing site. Then P site is vacating. While coming out, the already tryptophan tRNA with the tryptophan will comes on to the P site, and it is waiting for the another amino acid to form a polypeptide bond with this tryptophan. So here, when another amino acid is comes, that is A site, joins here, then it forms the bond with that amino acid, another amino acid, and then this tryptophan consisting tRNA molecule again existed, because this tryptophan is added into the polypeptide chain. It become free from the amino acid and it will exist. Step 3, translocation of ribosome along the mRNA from codon to codon. This helps to position the next codon in the S site. So now translocation. Here this site will not come their own. So once the polypeptide bond is formed between two amino acid, the ribosome will move from mRNA. Along with the mRNA, ribosome will move. That is translocation, moving of that ribosome along the mRNA from codon to codon. So then what happens? This helps the position the next codon in the amino acid. See here. we are mentioned with the EPA. So now once the amino acid comes and form the bond, the ribosome moves. As a result, the existed codon, the codon present in this existing region will comes out from the ribosome. Okay. Now, already we mentioned that EF elongation factors. There are TU and SU are the two types of elongation factors. So the joining of that amino acid, it requires GTP. So translocation means the translocation is occurred, translocation of ribosome, translocated. So means it changes its location. That means once the bond form, the ribosome will move along with the mRNA codon to codon. It moves. As a result, the existing size, that means site will comes out from the ribosome. So the process of elongation and peptide bond formation requires two factors, that is elongation factors, that is elongation factor TU and elongation factor TS. TU and TS, two factors are required. Termination of polypeptide chain, that is fourth step. Termination, that means stopping of the formation of polypeptide chain or maybe protein synthesis. The termination of polypeptide chain elongation takes place when termination or stop codons UAA, UAG, UGA, these are the stopping codons, enter the A site, because these stop codons do not have any tRNA molecule. The tRNA does not code for stop codons. Okay. So now these stop codons are recognized by release factors. tRNA is not respond to these stop codons. There is no tRNA for these stop codons, but these stop codons are recognized by the release factors, RF. So these relief factors are RF1 and RF2. RF1 terminates polypeptide chain in response to UAA, UAG, in response to these two codons. And RF2 terminates the polypeptide chain in response to UAA and UGA. Here it is difference. Both RF1 and RF2 factors response for UAA respond this both are respond to the UAA, but RF1 respond to the UAG and RF2 responds UGA. Here the difference is the RF1 is response is different here, this stop codon, and RF2 UGA. In prokaryotes, there are two release factors, while in eukaryotes, there is only one. So RF1 and RF2, two factors we can observe in prokaryotes, but whereas in eukaryotes, only one release factor to identify all these three stop codons. When the release factor in the site A or A site, when the release factor in A site, it alters the activity of peptidal transferase enzyme. So when release factor is present in site A, it alters, it changes the activity of peptidal transferase enzyme, and it adds the water molecule to the carboxyl terminus of the polypeptide chain. So generally, this polypeptide peptidal transferase enzyme adds the amino acid, but when the release factor is present, it changes that function as to add water molecule to the carboxyl terminus of the polypeptide chain. As a result, the polypeptide from the tRNA molecule in P site is released. So when mRNA molecule is released from the ribosome dissociates into subunits, separated into subunit. Termination is said to be complete. So now this stop codon, whenever this stop codon is appears, that is UAG, UAA or UGA. So there is no amino acid tRNA because these codons are not coding for any amino acid. Clear? Any amino acid. So as a result, what happens when this codon is present on this yellow color, it is A site. It is present on this region. Any of this codon present at this A site, it alters the function of that peptidal transferase enzyme. Generally, the peptidal transferase enzyme is helpful to form the bond between the amine group of amino acid as well as uh, carboxylic group of another amino acid to form the peptide bond. That is the function of the enzyme. But due to presence of the stop codon at this site A, it alters the function of that enzyme. So instead of bond between carboxylic acid and carboxylic group and amino group, it forms the bond between water molecule and carboxylic group. This terminates the polypeptide chain. It releases the polypeptide chain from the P site. Then it stops the extension of that polypeptide chain. And once the RNA is become free and polypeptide chain become free, both the subunits dissociates. Larger and smaller subunits are dissociates.